Hello guys, my name is Bing Wang. I'm a PhD student at Max Planck Institute for Human Cognitive and Brain Sciences. Today I'm going to talk about intracortical asymmetry in the human brain from postmortem cytoarchitecture to in vivo microstructure. Brain asymmetry is a quantitative index of interhemispheric differences between the contralateral homologous regions, and our brain is asymmetric to avoid duplication of neural circuitry that has equivalent functions, and that can be reflected in the brain structure and function. Regarding the brain structure, a large sample size Enigma study uses the healthy subjects to revive the cortical thickness and surface area asymmetry. Regarding the figure here, we can see the cortical thickness asymmetry pattern from left to right follows the anterior to posterior direction. And for the surface area asymmetry, the default motor network regions are more leftward asymmetry, shows leftward asymmetry, but for the frontal parietal cortices, they're showing the rightward asymmetry. And in addition to the surface area and the cortical thickness, the microstructure is a more specific measurement for the intracortical gray matter. And the Glasser in 2022, they revived the group averaged T1W, T2W ratio asymmetry in the whole brain. And we can see the left to right asymmetry is from the anterior to posterior direction. Also, regarding the cytoarchitecture results, Amans found that in the BA area 45, the left hemisphere intensity is higher than the right hemisphere intensity. Not only the microstructure measurement, but the microstructure profiles is also very organized in the human brain. And Casey Pacola just used the microstructure profile covariance, then we can see the correlation between the region A and the region B profiles and regress out the global intensity. They revived the microstructure organization of the principal components just from the anterior to posterior axis. So in the current literature, only the local postmortem microstructure asymmetry has been studied, and the group level large-scale in vivo microstructure asymmetry has also been studied, but there is no organization asymmetry studies. So to address the gaps, we want to answer the questions like how the individual effects on the large-scale microstructure asymmetry and whether the asymmetry is different in different layers, and how the intensity is organized in the right hemisphere and in the left hemisphere, and whether there is asymmetry in a globalized way. So we first used the big brain data, which has been described by Armin, and we used the 6-layer segmentation solution. And for the each layer, we reconstructed the 10 equal volumetric surfaces for each layer. So in total, there are 60 surfaces. And I summarized all the vertex data into the 180 puzzles using the multimodal puzzlation, and also summarized them into the CA12 networks. And for the figure B, we can see the asymmetry in each layers. And the asymmetry index is higher in the layer 6 and just smaller in the layer 1. And for the correlation between the asymmetry map of the asymmetry map between each layers, we can see the layer 4 shows the highest correlation within all the layers. And for the relative asymmetry, for example, the green color one, the language network, it differs between the layers. We can see the layer 1 just showed a very small proportion, and for the layer 4, it showed a very high proportion. And more specifically, I also showed the surface asymmetry along the intracortical dips by the network. And for example, the multimodal, the brown color line shows a correlation, increased correlation between the asymmetry and the intracortical dips, and as well as a language network. For the microstructure profile covariance gradients asymmetry, we used the intracortical intrahemispheric asymmetry and the interhemispheric asymmetry. Like for the example figure, the left hemisphere covariance and the right hemisphere covariance, and then we get the gradients and use left minus right, and I align the right left to the template of left gradients to make them comparable. And this result suggested the leftward asymmetry peaked in the inferior parietal cortices, and the rightward asymmetry peaked in the visual cortices along the first component, the anterior to posterior axis. And then we also use the in vivo data. We used the Mika mix and the HCP T1 T2 ratio, and the intensity in the Mika mix and HCP, they have a similar results, like high intensity highest in the sensory motor and the visual cortices, and the lowest intensity in the frontal areas. And the asymmetry of the intensity also follows anterior to posterior direction. And when I summarized that into 12 networks, it showed like the affective network, all the subjects showed the leftward asymmetry. And for the ventral multimodal network, almost the subjects showed the rightward asymmetry. And this results can be replicated in the HCP dataset. And for the gradients asymmetry used in vivo data, they also, the right left asymmetry also follows the anterior to posterior direction. And also the results can be replicated in the HCP dataset. So to conclude, in the present presentation just revived the postmortem intensity asymmetry pattern varies across layers, and postmortem cytoarchitecture organization shows leftward asymmetry in the inferior parietal gyrus and rightward asymmetry in the visual cortices along the anterior to posterior direction. The in vivo results suggested left-right intensity asymmetry follows an anterior to posterior direction, and the in vivo microstructure organization asymmetry also follows such the direction along the sensory fugal axis. And because we just revived the lateralization in the human structure, so we can apply our model to the disease sample like autism spectrum disorder, multimodal major depression disorder that is related to the lateralization dysfunction. And we are also explore how the intracortical asymmetry supports the functional asymmetry, and how third types interplay with the laminar pattern in asymmetry. And finally, we'll explore how the genetic and environmental factors shape the microstructure asymmetry. And I want to thank my co-authors and the data providers for their help. And thanks for listening. I'll take any questions.